Hello students, this video is to describe slide 34 to 55 under the topic of chemistry and functions of biomolecules. This lecture is focused only on amino acid and protein. Let's begin. Amino acid and protein is a nitrogen-containing biomolecules of which its basic structure containing amine and carboxylic groups linked by a chiral carbon atom at alpha position. The R side chain differ among others. There are hundreds of known amino acids in this world. However, there are only 20 amino acids are recognized as standard amino acid, which is mostly abundant in nature. Due to chirality, the stereoisomer-like monosaccharide D and L enantiomer occur. With L configuration mostly abundant in nature, there are four main groups of standard amino acid according to the chemical properties of the R side chain. Those are non-polar aliphatic R group, polar uncharged R group, polar charged R group, and aromatic R group. For non-polar aliphatic R group, it includes glycine, alanine, methionine, valine, leucine, and isoleucine. In this group, branch chain amino acid includes valine, leucine, and isoleucine. Glycine has no its enantiomer due to the lack of chirality. Methionine represents the thioester groups. The polar uncharged R group amino acid include serine and threonine containing hydroxyl groups with primary and secondary alcohol respectively. Cysteine has thiol group, asparagine and glutamine contains amide groups, while proline is a secondary amine amino acid. Another group is called polar charged R group, which can be either positively charged R group or negatively charged R group. The positively charged R group amino acid include lysine, arginine, and histidine. The negatively charged R group amino acid includes aspartate and glutamate due to the presence of carboxylic groups. The last group is called non-polar aromatic R group, includes phenylalanine containing benzyl functional group, tyrosine containing phenyl group, and tryptophan containing indole groups. Standard amino acid can be further classified as essential and non-essential amino acid, in which the former is unable to be synthesized in healthy human cells, while the latter is able to do so. There are 9 essential amino acids you might be familiar with: phenylalanine, lysine, methionine, histidine, tryptophan, isoleucine, leucine, threonine, and valine. The rest of them is of non-essential amino acid. Amino acid has its own properties to be deprotonated and protonated that result in negatively and positively charged within the molecule, so-called zwitterion or amphoteric molecule, in which it can act as a basic or acidic property in one molecule. This property makes amino acid has buffering properties, especially histidine, that is considered as the best buffer in physiological condition due to its pI is very close to physiological pH. Amino acid can condense to form peptides, oligopeptides, polypeptides, and protein. Using carboxylic group and one amine group from another, resulting in peptide bond between two amino acids. This action orients the peptide chain into N-terminal and C-terminal. We can call peptide derived from 2 units of amino acid as dipeptide, derived from 3 units of amino acid as tripeptide, so on and so forth. Let's practice with this peptide. Can you find the peptide bonds? It is C double bond O connected to nitrogen group. So there are 7 peptide bonds here. Therefore, there are 8 amino acids in this chain. So this peptide is known as octapeptide. Here are some examples of peptides. Aspartame is a dipeptide used as artificial sweetener instead of sugar. It is found in Diet Coke or Coke Zero that you may know. Glutathione is a tripeptide functioning as amino acid transport across membrane and cellular antioxidant against free radicals. Insulin, vasopressin, and oxytocin are peptide hormones to regulate our body function. Let's move on to protein. Protein is a polymer of amino acids, of which the units are greater than 100. Something between 50 to 100 amino acids is known as polypeptide, and something less than 50 amino acids is known as oligopeptides. Protein can be derived from only one chain of polypeptide called monomeric protein, and more than one polypeptide chain called multimeric protein. Protein has its specific structure in different levels. There are 4 levels of protein structure. Those are primary structure, secondary structure, tertiary structure, and quaternary structure. The multimeric protein can be recognized as homodimeric protein, in which there are 2 identical polypeptide chains are composed of, as heterodimeric protein, in which there are 2 different polypeptide chains are composed of. So can you guess what are these multimeric proteins are called? Of course, it is homotrimeric protein for this one, homotetameric protein for this one, and heterotetameric protein for this one. This figure shows the level of protein forming its structure from primary to quaternary structure. Primary structure is described as amino acid sequence of each protein, how the sequence order from N-terminal to C-terminal. The chemical bonds play a crucial role of this structure is only peptide bond. 2 identical proteins always show exactly similar amino acid sequence from N-terminal to C-terminal in every position. Let's have a look at the example here. Are they the same proteins? The answer is no. They are not the same protein. Even though both proteins have similar amino acid components, the sequence is totally different from N-terminal to C-terminal. Secondary structure is described as certain area with specific configurations resulted from intramolecular interaction among amino acids. Hydrogen bonds plays a crucial role to conform this level of protein structure. Alpha helix and beta pritide sheets are the result from the hydrogen bond. Helical structure and the pritide conformation at certain part of the protein chains are the features of secondary structure respectively. Beta sheet can be either in small sheet or large sheets, which is derived from parallel or antiparallel alignment of the chain. Parallel fashion is when hydrogen bond occurs between N-terminal to C-terminal and N-terminal to C-terminal chains, or between C-terminal and C to N terminal chain. In contrast, anti-parallel fashion is when hydrogen bond occurs between N terminal to C terminal and C terminal to N terminal chains, or between C to N and N to C terminal chains. Tertiary structure it describes as a 3D structure of a single whole polypeptide chain. There are many chemical interactions to support this level of structure within a single chain of polypeptide. Those are hydrogen bond, hydrophobic interaction between nonpolar R side chain amino acid, disulfide bond covalent bond between two cysteine residues, and ionic bonds between positively and negatively charged R side chain amino acid. You can see the tertiary structure of microbin and carbonic anhydrase, for example, which are monomeric protein. The monomeric protein have their structural level maximal as tertiary structure. In case of multimeric proteins, quaternary structure it describes as the conformation and interaction between among the chains in the protein. The classical examples are hemoglobin and immunoglobulin, containing 4 chains of polypeptides. The chemical bonds could resemble those of tertiary structure, but the interactions are in between the chains of polypeptide. Protein is easily denatured by many factors such as chemical reagent, temperature, mechanical force, and microbial activity. Denaturation can either be reversible or irreversible upon the removal of the denaturing factors. Once the native structure is changed, it results in loss of activity and the reduction in solubility and degradation resistance. In case of reversible denaturation, renaturation can occur when the denaturating agents are dissipated. So, the protein is back to fully function again. Denaturation is normally not resulted in the destruction of primary structure. In the other words, the peptide bonds are reserved for denaturation. However, when a protein undergoes proteolysis, it is referred to the destruction of peptide bonds at the primary structure, and the result from proteolysis yields peptides or amino acids. Protein has a lot of functions depending on types of protein. Here is the list of protein functions. Protein can be a structure of cells, tissue, and organ, such as collagen, keratin, and elastin. Protein can be a biological catalyst like enzymes, which you will learn deeply in another topic. Protein can act as a transporter like hemoglobin to transport oxygen and transferrin to transport iron. Ovalbumin is a storage protein for the growth of embryo, while the immunoglobulin and complements are protein to defend foreign particles. We know that the immunoglobulin could be called as antibodies. Protein, hormones, and growth factors function as regulators. Actin and myosin in muscle support cellular internal organ and the whole body movement. Finally, it can be metabolized for energy production in certain cases. Let's have a look. Albumin is the most protein found in serum or plasma of human blood.